Hello, I'm Funda Meric-Bernstam from MD Anderson Cancer Center. I'm excited to talk to you today about how to implement translational science in the clinic. Here are my disclosures. We talk about translational research a lot, and we talk about the T's. T0, referring to basic biomedical research, including preclinical and animal studies. T1, translating these findings to humans, including phase 1 clinical trials, where we assess the safety, tolerability of drugs, and get preliminary efficacy data. These are proof of concept studies. And T2 research, where we translate this into patients looking at efficacy. And finally, T3 research, translating into practice. And T4, translating into population health. Today, I'm going to mainly focus on T0, T1, T2, and with a focus on molecular cancer therapeutics. We talk a lot about molecular targeted therapy. The concept here is targeting changes in cancer cells that we believe help them grow, proliferate, survive, spread. Using these targeted therapies, informing the decisions using tumor and patient characteristics to select the optimal treatment is often referred to as precision oncology or personalized cancer therapy. When we talk about this molecular characterization of tumors, we can do this in a variety of ways. We talk a lot about genomics, looking for mutations, amplifications, deletions, or fusions, translocations. We can look at RNA, look at what gene expression changes occur, such as transcriptional profiling. We can look at protein levels. This is often referred to as proteomic profiling. Or we can look at different changes, such as methylation of DNA or tumor metabolism, and very recently, a lot of excitement around immune profiling. Several studies have been done recently looking at a large number of cholangiocarcinoma tumor samples, trying to identify differences between these tumors and trying to identify what are the differences that occur in the tumor compared to normal. The goals of these studies to understand the drivers of cholangiocarcinoma. Now, some of these studies have shown that depending on the etiology, for example, fluke-related cholangiocarcinomas versus non-fluke-related cholangiocarcinomas may have differences at the molecular level, and thus they may have different drivers. And this is very helpful to do these integrated analyses, looking at changes in the copy number of genes, looking at specific mutations, looking at expression, DNA methylation, to try to understand, can we understand cholangiocarcinoma better? And indeed, looking at these differences, we see that actually these can be different classifications and different categories of cholangiocarcinoma not only may have different drivers, but in fact may be different prognostically. That's important, of course, because in clinical trials, if we're selecting a certain number of patients or certain tumor alterations, it's important to know that these different tumor alterations may have different biology and different behavior. A lot of attention has been on genetic alterations, specifically mutations that drive cholangiocarcinoma. And we have seen now, exciting activity with FGFR alterations, especially FGFR fusions, and early promise with HER2 alterations, and then several other alterations that we've been aware of in the MAP kinase signaling pathway that previously were not actionable because we didn't really have drugs that were efficacious in space. But there's now a lot of trials that are ongoing that are starting to look at these alterations. And I hope over the next few years, we're really going to have therapeutic options. These kind of clinical trials require actually organized efforts. However, cholangiocarcinoma being rare, it's actually hard to do these trials that are referred to, for example, as umbrella trials, where you test a certain tumor type with cholangiocarcinoma and give different treatments based on the tumor type, based on molecular alterations. However, there's been a lot of momentum also gained through trials referred to as basket trials. These are trials looking at alterations that are rare, but they're found across a variety of tumor types. So even patients that have rare tumors, such as cholangiocarcinoma, may benefit by going on these trials if they have an appropriate molecular alteration. Implementing these kind of trials is actually very important to have different treatment options for patients that are undergoing genomic testing, but also infrastructure is needed to best match treatments to patients. And at Anderson, we've done this through putting together a research program and a precision oncology decision support effort. This works as a core. We have a group of research scientists interested in oncology and a group of oncologists interested in genomics working together and trying to put together best associations for treatment options. And this has led to a precision oncology decision support knowledge base. It has allowed us to review what's known in the literature to be a genomic alteration, a genomic variant or a copy number change or a fusion that's actionable. It's allowed us to classify drugs based on their targets, classify trials based on what kind of patients, what kind of alterations they're looking for, and put it into context based on level of evidence about what genomic alterations worth acting on, and how do we compare that based on standard options. This has allowed us for each individual patient to generate patient reports to interpret genomic alterations, whether the testing is done at MD Anderson or elsewhere. It also allowed us to build a publicly available website, personalizedcancertherapy.org. And for physicians, we also have a searchable clinical trial portal so they can identify trials that are relevant for their patients. And we've also initiated a clinical trial alert system that I can tell you about. Here, what we do is when patients undergo genomic testing, the treating physician gets an alert saying your patient has an actionable alteration and the following clinical trials may be relevant. On top of that, directed to the researchers, when they activate a trial, they're informed of patients that have molecularly tumors that are eligible. And further, every day or every week, they get an update saying a patient has been diagnosed with an alteration in their tumor that may be a good match for your trial. These kind of efforts really have fueled enrollment onto basket trials as well as umbrella trials. And we've already started seeing, efficacy signal in some of these trials, as you know. Some examples of genomically relevant targets that seem very promising include FGFR, which, as you know, is very exciting because this is the first target that's genomically driven that has now FDA approval. And in addition to the FDA-approved pemigatinib that is really has had elegant data for cholangiocarcinoma with FGFR fusions, there's been several other agents that have significant efficacy signal with FGFR inhibition for FGFR fusions, really confirming that FGFR fusions indeed are a compelling target for cholangiocarcinoma. Another target that's been interesting is BRAF. As you know, BRAF V600 altered in melanoma and really transformed the outcomes in melanoma patients that have a BRAF V600E mutation with the approval of first RAF inhibitors, then RAF plus MEK inhibitors. Now, there's subsequently been approval of RAF or BRAF or RAF plus MEK inhibitors in other disease types, including Erdheim-Chester disease, anaplastic thyroid cancer, as well as BRAF mutant lung cancer. It's important to note in BRAF V600E mutant biliary tumors, there's also really compelling efficacy signal with dabrafenib trametinib shown here. However, yet this is not an approved FDA approved therapeutic option, but clearly I think a target when identified in a patient report that's worth pursuing. Another really compelling target in my mind across tumor types is HER2. And this is a very exciting space because many different treatment options is evolving for HER2. We've shown at MD Anderson that patients that have HER2 positive disease that don't have breast or gastric cancer where HER2-targeted therapy is already approved. If they get HER2-targeted therapy, they have a substantial improvement in their progression-free survival, with a trend of improvement in overall survival, even though this was a relatively small set of patients. And you can see some of these targeted therapies are really showing promise in biliary cancer as well. So here is a combination of pertuzumab and trastuzumab in patients treated on a Basque trial, referred to as My Pathway. And you can see the efficacy signal in biliary cancer in this cohort. This is early data. Another clinical trial ongoing with a bispecific HER2 inhibitor, ZW25. This trial is accruing HER2-positive patients with a variety of different tumor types. In salmon are the patients that are HER2-positive and have biliary cancer. And you can see, as on this waterfall plot, really compelling responses seen in this tumor type. Dr. Subhan Pant at this meeting will be talking a little bit more about this trial data. Another really exciting agent is trastuzumab deruxtecan. This is an antibody-drug conjugate, so it's an antibody that attaches HER2 with a chemotherapy payload. This drug recently was approved for HER2-positive breast cancer and is now being tested for HER2-positive other tumor types. So these kind of clinical signals and early clinical trials are very exciting, and these trials, of course, are working to optimize dose and schedule so we can target this critical pathway while minimizing normal tissue toxicity. But it's important to note that not all patients respond, and responses often, unfortunately, are not durable. So as translational researchers, we not only want to enroll on these trials, but we want to use these trials as opportunities to better understand the drugs. We want to understand the mechanisms of resistance, whether it was just because the tumor was heterogeneous, whether some of these tumors were intrinsically resistant, what are the adaptive responses in the tumor, meaning when you treat the tumor, what does the tumor do to overcome the drug, and what are the acquired resistance mutations. I want to point out that for these kind of strategies, a key component is research biopsies. And in a place like Cholangiocarcinoma Foundation, I have to really ask for some partnership with our advocacy groups in this space. Many clinical trials have research biopsies built in, and it's a little elusive, so let me just clarify that in some trials, the biopsies are mandatory. They're required to conduct the study, and only patients that enroll, and agree to get a biopsy can be enrolled in the trial. The biopsy is going to be a very important endpoint in the study. Some other trials have optional biopsies. The biopsy is not required for enrollment, but these are usually incorporated because the biopsy is likely to reveal very important scientific information that can help not only develop the drug better, but help select patients better for the next generation of trials. When we talk about research biopsies, there are a few different kinds of biopsies, and I just want to tell you a little bit more about why we do them. So pre-treatment biopsies are done prior to starting the treatment. This is sometimes required because we want to know if the target's present. Sometimes it's required to determine eligibility on a trial. For example, some HER2-targeted therapy trials may require testing HER2. In other scenarios, it's used to help identify predictive markers. The goal is to identify biomarkers that can help determine which patients will respond, which patients will not respond for therapy. On-treatment biopsies done during treatment are used to determine whether the target of the drug is being inhibited and what the tumor does in response to that inhibition. And then patients that respond to treatment but ultimately progress may be approached to get a post-progression of the biopsy. This is so that we can learn better why the tumors get resistant. Sometimes we can use that same biopsy to learn more about the tumor genomics in a clinical lab so we can help with selecting the next treatment choice as well. Another thing that we know is some of these genomic information can be ascertained by doing liquid biopsies where we can check these DNA changes in the plasma. So patients may be approached to get additional plasma samples throughout their treatment. And again, all of these I think are really very important scientifically for us to develop drugs better. Another challenge is really the preclinical research, the T0 translation that we talk about. In cholangiocarcinoma especially, we have very few resources. We don't have that many cell lines. We don't have that many patient-derived organoids. And we don't have that many in vivo models. So I think this is an area that we really need to partner on as a group to try to generate new models. An area that we focused on at MD Anderson is developing patient-derived models from cholangiocarcinoma. Here we take tumor samples from patients either obtained at the time of surgery or obtained with an extra biopsy when they're undergoing a biopsy already, so that we can capture a little bit more information about different tumor types and how they respond to different therapies. This allows us to test different targeted therapies or non-targeted therapies, as well as understand better how the tumor responds, how it becomes resistant, and what are the best combinations. This is layered on some other programs, for example, our unusual responder program, where we systematically approach patients that are undergoing certain targeted therapies to see if we can get an additional sample before we start treatment and a sample at the end of treatment along with plasma collection throughout, so we can better understand how tumor evolves. And finally, our MD Anderson Rare Tumor Initiative, where we've looked across tumor types, and cholangiocarcinoma was picked as one of the most relevant tumors where we really want to make an impact by doing extensive molecular profiling to create greater insight. You can tell from all these efforts that the key aspect in translational research, in fact, is another T I haven't mentioned, which is teamwork. I've already talked a little bit about our cholangiocarcinoma team that has worked together in research. I also want to acknowledge also the Institute of Personalized Cancer Therapy, the decision support team, and our Department of Investigation Cancer Therapeutics. And thank you for your attention.
Hello, good morning. Thanks for inviting me to give this talk here. Thank you for organizing this meeting in this virtual meeting in this special moment for everyone that we are living all around the world. I'm Patricia Aspichueta from the Lipids and Liver Group from the University of the Basque Country, Faculty of Medicine. As you know, metabolism is at the basis of all biological activities. Metabolic pathways support cell survival, are highly interconnected and coordinated roads, are dynamic roads in continuous movement, and this makes it so complex to investigate. In physiological conditions such as fasting, feeding, refeeding, exercise, metabolic rewiring will take place to adjust to the new environment, and homeostasis will soon be recovered. However, in pathological situations such as in cancer, in which the driving cause won't be removed, the metabolic rewiring will affect one and some other metabolic pathways, and as the disease progresses, new causes of metabolic reprogramming will also appear. In cancer cells, there is a high increased of uptake of precursors for the synthesis of molecules that will allow the accumulation of biomass during cellular proliferation. In cancer cells, the increased uptake of glucose will support the active glycolysis, in which intermediates will be used for the pentose phosphate pathways, for the generation of glycerol 3-phosphate, or for 1-carbon metabolism. At the same time, in these metabolic pathways, intermediates will be used as precursors for molecules such as lipids. The TCA cycle is a source of FADH2 and NADH. In cancer cells, the high uptake of glutamine will feed the TCA cycle, where intermediates will also be generated, and some, such as citrate, will be transported to the cytosol to generate acetyl-CoA. At the same time, we will have other sources of acetyl-CoA coming into the cell. As you all know, the acetyl-CoA is one of the most important molecules in energetic metabolism, and among others, will also participate in the synthesis of new fatty acids and cholesterol into the cells. So, rewiring of lipid metabolism and lipids are required in cancer cells. We know that glycerolipids, esterols are required for the generation of new cellular membranes, that TGs, triglycerides are energy storage forms, and that lipids such as ceramides and sphingolipids are cell signaling molecules required for cancer cells. So, targeting metabolic pathways involved in lipid metabolism could provide new therapeutic targets. More investigation and more research has been performed in the hepatocellular carcinoma cancer type. In fact, we know that there are ongoing clinical trials with molecule drugs that inhibit metabolic pathways involved in lipid metabolism so that recurrence of ACC could be prevented. We need to gain knowledge into the rewiring of metabolism in cholangiocarcinoma. The last years, there have been an active research in this area. So we know now that cholangiocarcinoma cells also require high amount of precursors for the generation of new molecules. In fact, we know that the glucose transporter GLUT1 is upregulated in cholangiocarcinoma, and that it correlates with increased glucose uptake and with the tumor status and tumor size. Besides, in cholangiocarcinoma, it has been described that GLUT1 positive tumors have a poorer disease-related survival. Concerning the role of glutamine in the TCA cycle, we still have a lot to investigate. However, recently it has been established that glutamine levels are strongly decreased in cholangiocarcinoma samples, and the growth of human cholangiocarcinoma cell lines is highly dependent on glutamine. This suggests that glutamine is also feeding the TCA cycle to generate intermediates, precursors for the synthesis of all of the molecules that are required in cholangiocarcinoma cells. The isofiltrate dehydrogenase is an enzyme that is involved in the TCA cycle, and there are several genes that present polymorphisms in IDH, and this is the case for some patients with intrahepatic cholangiocarcinoma. The polymorphisms or mutations in this IDH have been linked to the generation of an oncometabolite that will promote the progression of the disease. Less is known about lipid metabolism and cholangiocarcinoma. However, it has been recently established that fatty acid uptake in intrahepatic cholangiocarcinoma cells is comparable to that of normal hepatocytes, in which already the uptake is higher than in other cell types. Besides, it has been established that lipoprotein deficient media strongly inhibits self-growth of an intrahepatic cholangiocarcinoma cell line. These results suggest that cholangiocarcinoma cells required lipids coming from extracellular sources. This is supported by the fact that it has been described that enzymes involved in the novel lipogenesis of lipids inside the cells are decreased in intrahepatic cholangiocarcinoma tumors when comparing to the surrounding tissue. Besides, it has been described that fatty acid synthase, FAS, is dispensable for the intrahepatic cholangiocarcinoma development in mice. We have recently performed some experiments in our lab, these are unpublished data, and we have found that in the intrahepatic and the extrahepatic cholangiocarcinoma cell lines, the uptake of the fatty acid oleate is increased. Concerning lipoprotein uptake, we have found that the lipoproteins VLDL and HDL uptake is increased in the extrahepatic cholangiocarcinoma cell line, while it's not in the intrahepatic cholangiocarcinoma cell line. However, the LDL uptake is decreased in the intrahepatic cholangiocarcinoma cell line. So next we wanted to know what would be the metabolic fate of these fatty acids. So we measured triglyceride synthesis. We found that in intrahepatic and extrahepatic cholangiocarcinoma cell lines, the TG synthesis was increased. We found the same result when we used tissues from a generated model of intrahepatic cholangiocarcinoma. TG synthesis was also increased. In both cases, we found that this increased TG synthesis led to an accumulation of triglycerides in cell lines and in livers. Curiously, when we observed that the TG synthesis was higher in the extrahepatic cholangiocarcinoma cell line than in the intrahepatic one, the TG content was increased to a higher amount in the intrahepatic cholangiocarcinoma cell line. So we proposed that the fatty acid oxidation rate will be differently in both cholangiocarcinoma cell lines. And indeed we found that in the intrahepatic cholangiocarcinoma cell lines, probably as a result of the decreased fatty acid oxidation rate, the accumulation of this TG concentration was taking place. However, in the extrahepatic cholangiocarcinoma cell line, we found a high increase in the fatty acid oxidation rate. These results tells us that the intrahepatic and the extrahepatic cholangiocarcinoma cell lines relies differently on fatty acid oxidation rate. As in the intrahepatic cholangiocarcinoma cell lines, the results showed us that in the mouse model of intrahepatic cholangiocarcinoma, there was also a decrease in the fatty acid oxidation rate together with levels of CPT2. CPT2 is involved in the channeling of fatty acids from the cytosol to the mitochondria. So the next question is the metabolic rewiring a cause or a consequence of cancer? So may changes in signaling pathways rewire metabolism? So this has been investigated for hepatocellular carcinoma, where levels of fatty acid synthase are increased. Calvisi group found that while fatty acid synthesis is required for AKT and AKT-c-MET driving hepatocarcinogenesis, is dispensable for c-MET and beta-catenin-induced liver cancer, which this tells us is depending on the cell line that will be in the signaling pathway that will be activated. The requirement for one or other metabolic pathway will be different. We don't know if this is happening also in cholangiocarcinoma. The next question is, how is the environment changing the metabolic rewiring or the reprogramming of metabolism? So we know that in non-alcoholic fatty liver disease, there is a really lipid-rich environment. Free fatty acids, lipoprotein levels are increased. And we also know that in NAFLD-related HCC, there is a downregulation of CPT2 and as a consequence, a decreased fatty acid oxidation rate. So this lipid-rich environment will induce the remodeling in metabolism. As a consequence of this CPT2 downregulation, there is an accumulation of acylcarnitines, which will activate STAT3. So in this case, it's the environment, the one that it's inducing directly changes in the metabolic reprogramming. Well, this is of particularly interest because we are talking about the liver, and the liver is a central organ controlling whole body metabolic homeostasis. The liver is exposed to different signals coming from the brain, the adipose tissue, and all these signals will modulate metabolic pathways in liver to synthesize compounds to give response to the whole body metabolic requirements. But in obesity, in the metabolic syndrome, when there is an insulin resistance, some of these signals will be disturbed, and as a consequence also the different metabolic pathways and the generation of different compounds, leading to non-alcoholic fatty liver disease. Non-alcoholic fatty liver disease starts with the accumulation of lipids, but it may progress in some patients to NASH or non-alcoholic steatohepatitis, cirrhosis, and or hepatocellular carcinoma. The prevalence of NAFLD is rising its day because mainly the metabolic diseases are also increasing. NAFLD is increasing with a high rate of increase in obesity and diabetes. NAFLD has been associated with nearly twofold increase in the risk of developing cancers predominantly in liver, and is associated with intrahepatic cholangiocarcinoma more than to extrahepatic cholangiocarcinoma. The NAFLD pathogenesis starts as a misbalance between pathways that control lipid input and lipid output in liver. As a consequence, new compounds, lipotoxicity coming from those compounds, new metabolites, cytokines, hepatokines will be released, leading to a more complicated metabolic scenario. And this is the case for osteopontin. Osteopontin, we know that it's increased in serum of non-alcoholic fatty liver disease patients. We, 2 years ago, found that the osteopontin levels in serum correlates with intrahepatic concentration of phosphatidylcholine, phosphatidylethanolamine, and cholesterol. But we also know that osteopontin is the most overexpressed gene in intrahepatic cholangiocarcinoma, and that osteopontin levels are increased in serum of cholangiocarcinoma patients. So we went ahead and we also found that circulating osteopontin in serum controls the liver PC, the phosphatidylcholine, and phosphatidylethanolamine levels. So osteopontin in serum controls lipid metabolism in liver. Not only this, but osteopontin in serum also activates the FAC and the AKT signaling pathways which are involved in cholangiocarcinoma. So these results may suggest that somehow osteopontin could act as a link between NAFLD and cholangiocarcinoma development. So as a take-home message, I would say a hallmark of liver cancer is the rewiring of lipid metabolism. Cholangiocarcinoma cells require high amounts of precursors, including lipids, for the synthesis of molecules that will allow proliferation. In cholangiocarcinoma cells, uptake of fatty acids as free or into lipoproteins is increased. In cholangiocarcinoma cells, there is a dysregulation in glycerolipid synthesis and in fatty acid oxidation rate. The genetics and the environment are independent contributors to a metabolic dysregulation of liver cancer, and NAFLD is a risk factor for liver cancers, including intrahepatic cholangiocarcinoma. So I would like to thank my group, the Lipids and Liver Group. I would like to thank also to people from the University of Basque Country, like Ana Zubiaga and Cesar Martin. I would also like to thank people from beyond Donosti Health Research Institute, like the group of Jesus Bañales, Pedro Rodriguez, Machus Perugorria, and also I would like to thank Diego Calvisi, who has provided the tools to develop the animal models of intrahepatic cholangiocarcinoma. So thank you very much for your attention.
Hello, I am Rocio Macias from the University of Salamanca in Spain. This is my first Cholangiocarcinoma Foundation meeting, and I was looking forward to visiting Salt Lake, but I hope I will have another opportunity in the future. I want to thank the organizers for the invitation to participate in this special conference and the CCF team for the great work and support organizing this online conference. My talk is about drug resistance in cholangiocarcinoma. I think it's a concept you all know, but I hope I can offer you new information. So, what is drug resistance? I ask Stacy's permission to use this tumor cell you are familiar with to illustrate what happens in anti-cancer drug resistance. Resistance occurs when tumor cells don't respond to treatment with anti-cancer drugs. This is a problem that occurs with classical chemotherapy, but also with targeted therapy and with immunotherapy. And it's a clinical challenge since some drugs that have a certain effect reducing the growth in other solid tumors are not so effective in cholangiocarcinomas because biliary tumors are highly resistant. Another thing you know is that cholangiocarcinomas are characterized by heterogeneity. When we compare tumors from different patients, we can see important differences, but also in one tumor, we can have different cells with different characteristics, and this explains partially drug resistance. Resistance is known as primary or intrinsic when it's present before the administration of any pharmacological treatment. As I said, it exists genetic diversity among cells within a tumor, and some cells can be resistant to a therapy before they are ever exposed. When a tumor is treated with a certain drug, susceptible cells die and the tumor shrinks, but resistant cells survive and the tumor regrows after the treatment. The more heterogeneous a tumor is, the more likely some cells are to be resistant. But tumors can develop resistance after exposure to a therapy. That is, there is an initial response followed by loss of efficacy, which is known as secondary or acquired resistance. Even if all the cells are the same, which is the case of a cell line or a tumor that grows from a cell line in experimental conditions, changes in some genes or mutations can occur when cells try to defend themselves against the drug. So the cells that survive will form new tumors that don't respond to this specific drug. In addition, in some cases, the mechanisms that tumor cells use to defend themselves against one drug can be effective against other drugs. This is known as cross-resistance or multi-drug resistance phenotype. In this case, the tumor formed by resistant cells continue to grow despite treatment with different drugs that the cells can fight with the same weapons. Well, let's revise the mechanisms of chemoresistance or MOGs. We have divided them into 7 groups. MOG1 involves transport proteins. Most anti-cancer agents have their molecular targets inside tumor cells, but due to their structure or physical chemical properties, they cannot cross the plasma membrane by simple diffusion, but they need to use specific transport proteins as doors of entry into the cells. If some doors are blocked, we will have less drug inside the cells and less effect. But even if the entry functions normally, the cells have another strategy to reduce the amount of drug. They use a specific transporter that export the drugs, that eliminate the drug from the cell, and this is a very common mechanism of chemoresistance, ATP binding cassette or ABC proteins. In cholangiocarcinoma, reduced levels of the nucleoside transporter 1 is involved in lower response to gemcitabine, and decreased levels of OCT1, the organic cation transporter 1, explain lower effect of cisplatin and sorafenib. And increased levels of these export pumps are responsible of lower effect of different drugs. MOG2 is associated with drug metabolism. The cells have enzymes that participated in the detoxification of potential toxic compounds, and tumor cells can use these enzymes to inactivate drug. So we will have less effect if we have a less amount of these enzymes. But we have to keep in mind that in some cases, drugs are administered as prodrugs, such as gemcitabine. That means that they need to be activated inside the tumor cells. And the contrary, if we have less enzymes, we will have less active drug and less effect. And it's important that this this prodrug is not inactivated before obtaining the active drug. So in cholangiocarcinoma, increased levels of these enzymes are associated with lower response to gemcitabine and cisplatin. MOG3 refers to changes in molecular targets. These targets can be inside the tumor cells or can be in the plasma membrane. But in any case, changes in the expression or in the functionality of these molecular targets can reduce the effect of drugs. For instance, increased levels of thymidylate synthase explain a lower effect of 5-fluorouracil, and increased levels of epidermal growth factor receptor occurs and is associated with lower response to erlotinib. MOG4 only affect drugs that bind to DNA. Tumor cells are able to detect a damage in the DNA and induce changes to repair this alteration. If we have increased levels of enzymes that participate in the repair machinery, we will have less effects of this type of drugs. For instance, increased levels of these enzymes predict lower response to cisplatin and gemcitabine. MOG5 include changes in the balance apoptosis survival. Here we have a high number of genes whose change affect survival pathways. If we have lower levels of genes involved in apoptosis or increased levels of genes that block apoptosis, we will have less effect of chemotherapy. MOG6 is involved in microenvironment. The stromal tissue surrounding tumor cells is a barrier that makes it difficult for drugs to reach the tumor cells, but the components of this microenvironment produce changes such as hypoxia or acidification of the extracellular medium, or produce signals such as cytokines that affect the response of the cells to chemotherapy. For instance, acidification of the medium and hypoxia have been found in tumors that don't respond to gemcitabine. And finally, MOG7 refers to enhanced phenotype transition. Mesenchymal cells, that means this change from epithelial to mesenchymal transition. And mesenchymal cells are considered highly chemoresistant. For instance, increased levels of these proteins are associated with lower response to gemcitabine by inducing this epithelial mesenchymal transition. We have to keep in mind that more than one mechanism can contribute to this drug resistance at the same time. And also, this is not a fixed situation, but a dynamic situation. It's very important to know more about the mechanisms of drug resistance because we know that the same treatment is not the best option for all the patients. And all the information maybe can be useful in the future for clinicians to help to select the best option for patients, because we know a priori that some patients will not respond and some will relapse after some time. We have investigated a high number of genes involved in drug resistance in cholangiocarcinoma, and based on our experience, we have selected around 100 that we can measure by Tagman low-density array. We are analyzing the expression of genes in tumors from patients that are treated with GMCs, and we have seen that in basal conditions we have a very different expression depending on the genes from one patient to the other. We have the information of the radiological response, and I hope by the end of this year we will have the results of this study and we will have some conclusions. In our group, we are especially interested in transport proteins, and we use different models from less to more complex to investigate different aspects of chemoresistance. For instance, it's possible to investigate the contribution of transport proteins for each drug using frog oocytes or cell lines expressing specific transport proteins. Using this model of frog oocytes, we have demonstrated oocytes that express OCT1. We have demonstrated that this transport protein is able to take up sorafenib. We confirm this using cell lines expressing OCT1, and in another tumor, in another hepatic tumor, we found that it was frequent the presence of mutations that gave non-functional proteins with OCT1 located inside the cells instead that in the plasma membrane. In a collaborative study with a German group, we found that the expression of OCT1 was very different from one tumor to others. And we observed that there was no differences in the overall survival of patients treated with sorafenib if we just compare the expression or absence of OCT1. However, overall expression was higher in patients whose tumors had OCT1 expressed in the plasma membrane. Sorafenib is not used in cholangiocarcinoma because the first studies showed no beneficial effect. And in a study in different cohorts, we showed that the expression of OCT1 is very low in cholangiocarcinomas. And probably this explains, at least partially, why these tumors don't respond to sorafenib. In fact, when we include OCT1 in cell lines, we demonstrated both in vitro and in vivo that sorafenib was effective, reducing cell viability and the size of the tumors. Okay, these cholangiocarcinomas don't have OCT1, and this affects the activity of cisplatin, but we have seen that cholangiocarcinomas express other transport proteins. For instance, ASBT, a transporter that permits the uptake of bile acids, is present in cholangiocyte and it is also present in cholangiocarcinomas. Both the levels are similar than in surrounding non-tumoral tissue and is located in the plasma membrane. So we have used this characteristic to treat tumor cells and send more cisplatin to these cholangiocarcinoma cells. What we have done is to synthesize a family of compounds by binding cisplatin to bile acids. Cisplatin is hidden in the tract, and we have sent this compound to the tumor. When we compare the effect of BAMETs with cisplatin, we found that the amount of drug in the tumor was higher than when we administered cisplatin, and also the anti-tumoral effect was better. And the toxic effect was less because BAMETs are maintained in the intrahepatic circulation. So it's very important to know more about the mechanisms of chemoresistance for each drug in cholangiocarcinoma, including the presence or absence or transport protein in the plasma membrane, because we can select the best treatment for each patient at each moment. And also we can design personalized strategies to overcome drug resistance. We are playing a particular molecular chess game against cholangiocarcinoma, and all the information we can obtain can be useful to be at least one step ahead of the tumor to decide the best next movement. In this field, the research is already focused on personalized medicine to select the best treatment for each patient, in designing better drugs, more potent, or that are able to escape resistance, design better combinatorial treatments to fight the tumor from different sides, the identification of new targets for drugs, and the design of strategies to overcome drug resistance. And with this rare and heterogeneous tumor, collaborative work is very important. And ENSCA and also Cholangiocarcinoma Foundation play a very important role to work fast. Finally, I want to thank my group and all collaborators, and also I want to thank you for joining. Thank you.
Hello, everybody. Welcome to the panel discussion. I first want to, I'm Sergio Gradilone from the Hormel Institute at the University of Minnesota. First, I want to thank the organizers for putting together this fantastic conference. This is a unique opportunity, mainly for basic scientists. Having this interaction with patients and caregivers is really very important for us. So we just have a very interesting session with a very touching story by Jessica. Thank you so much, Jessica, for sharing that with us. And a very interesting scientific program with 3 presentations highlighting the importance of basic research and translational research. So we have with us Funda, that is on the phone. We have Rocio and Patricia. I think Patricia is also on the phone. So please feel free to start asking questions on the chat for our speakers. So I will start with a few questions that, well, actually several questions that we have from the audience. So Funda, I hope you are listening to us. One of the persons in the audience, Sehal Shah, asked, he's asking what research can be done to determine the causes of cholangiocarcinoma. He's saying so many people with healthy lifestyles get this cancer. What could be the causes of this cancer? Do you want to comment in general on this?
Yeah, this is a hard question, of course, not just for cholangiocarcinoma, but in fact for all cancers. You know, we know that cancer can develop in the context of a variety of factors, including some environmental factors, things like, you know, fluke-related infections that are cause inflammation and linked to cholangiocarcinoma development. There are some genetic factors where we know that there may be some predisposition linked to some genetic alterations, but predominantly, you know, we don't have a culprit and not, you know, something we can pinpoint as a cause of cancer. That is where I think translational research comes into play, where we can better characterize tumors in patients, you know, in fact, across different countries, across the different age groups and ethnicities, to understand what is the molecular makeup of each tumor, and can we identify the cancer drivers that we can then more effectively use for therapy targeting.
Thank you.
Yeah, and besides, I'm Patricia. And besides this, we need to take into account that also these multifactorial causes may change during the progression of the disease. So we need to follow up, because maybe, for example, the metabolic side will change depending on what signaling pathways will get activated through the progression of the disease. So it's the cause and also how it progresses, what may change also.
So it's a second question for Funda regarding translational research. The question is if biopsies as are practiced capture tumor heterogeneity.
That's an excellent question. And we do know heterogeneity has a lot of aspects. So, you know, we know that in cholangiocarcinoma, many of our patients, unfortunately, are diagnosed at a time where tumor may have spread. So our diagnosis is made with a biopsy, usually a needle biopsy. So that's really a fraction of the tumor, of course, that the patient has. The thought is that the driving alterations may be in most tumors, but we know that as we give treatment and as the tumor spreads, additional changes may occur. So thus, we're actively looking to see what is the value added of repeat biopsies, especially if you get a targeted therapy, and we know that now there's mechanisms of resistance with new mutations or other changes that can confer resistance. Again, another area that's being actively explored is liquid biopsies. Can we look in the plasma for example, for changes in the DNA that starts occurring. And indeed, we know that, even, for example, in a patient with an FGFR fusion, if we give FGFR inhibitor, there may be different mutations that start occurring in different areas within the same tumor. So I agree that heterogeneity, the differences in different areas of the tumor, adds an extra layer of challenge, emphasizing to us that I think we should try to move the most effective treatments up and try to do the best job as we can in the first line when we identify these, these tumors.
Thank you, Funda. I have here a question for Patricia. Thank you so much for your presentation, Patricia. So the question is a little bit on how this metabolic reprogramming is regulated. Specifically, the question is, what drives this metabolic rewiring in liver tumors? Is this genetics or these are environmental reasons? What's your take on that?
Well, the thing is that it's a multifactorial cause. It may, in some cases, the genetics has a lot to do. We know that mutations in several genes, such as P53, leads to a new metabolic scenario. But the thing is that also the environment and also the, for example, if we focus on lipids, the fat accumulation in different tissues will have a lot to do also, because this fat accumulation may induce mutations and also may change signaling pathways that at the end will lead to proliferation. So we need to take into consideration that it's not just a liver disease, it's a whole body disease, and that as other cancers, they need energy supplies. So we need to know in each type which kind or what metabolic pathways are the ones that are taking into or are more more involved in this subtype. So I would say that it's a combination, and we need to stratify also from the metabolic point of view, and I think that it could help a lot in therapies, and we need to take into consideration the diet, nutrition, and this also could help.
Thank you. Another question for you, Patricia. This is coming from Sehal and Karen. The idea here is, I saw that you already comment something on the chat. So they are asking if there are any ongoing studies in the clinic for the intervention of lipid metabolism, if there are any repurposed drug that we can use in cholangiocarcinoma.
Yeah. Well, for hepatocellular carcinoma, there are ongoing trials, but as far as I know, not for cholangiocarcinoma, not the typical drugs that are being used for metabolic diseases, but each day we get more research, or ongoing research focused on this. And for example, recently it has been discovered that PPAR alpha activation, which will promote the activation of this usage of energy, will also be involved in the proliferation of cholangiocarcinoma. So and there are different systems to inhibit this PPAR alpha activation. So this is something that we need to take into consideration, yes.
Thank you. Rocio, thank you so much for your talk. I have a question here from Jesus Bañales. He said that clinical trials are very much focused on therapeutic targets, but do not take into account the baseline and acquired chemoresistant mechanism of tumor cells. How we could improve it? Could we analyze circulating DNA mutations related to transport of drugs?
Okay, I see like several questions. I agree that when a drug is selected because a patient has a certain characteristic, they just take into account the presence of absence or a mutation to decide that this is good. But sometimes even these targets are located in the plasma membrane, but the drugs need to enter the cells to have an effect. So maybe to take into account if the drug can have more facilities to be inside or to be metabolized could help to know if we are going to have an effect or not, because we see that patients with the same mutation, some have an effect and other less effect. So there are other things that are also affecting the response. And regarding if it is possible to check in blood if we have like mutations or something related to drug resistance, I'm sure in the future we will have analysis to predict the response to drug without needing a tumor biopsy. Some things can be done now, but I think in the future we will have some improvements.
Thank you. Rocio, can you comment a little bit on this nice strategy you show us about binding cisplatin to a bile acid to get it transported through ASVT? What do you expect any off-target effects if this bile acid is getting in different cells, different organs? What do you expect from that?
We have tried that for hepatocellular carcinoma because bile acids will also go to hepatocytes and intestinal cells because they have transport proteins that permits that these molecules are located in the enterohepatic circulation. But we have done some experiments, and these molecules are less toxic because you have less cisplatin in the organism, in the circulation. You have the drug concentrated in the enterohepatic circulation, and even if you have this ASVT in these cells, they have other molecules to export bile acids. So also to export these bile acids with cisplatin, while the tumor cells in some cases have less capacity than hepatocytes or ileocytes, or cholangiocytes. So in the studies we have done, we don't have toxic effects in other cells, and the effect of BAMETs it's better than cisplatin alone.
Thank you. We have a question here from Juan Valle. I guess that probably Funda can comment on this. It's related to a previous question. Can the speaker comment on the relevance of a single biopsy on which treatment decisions are made when considering heterogeneity and resistance? Should we be starting sampling multiple areas of the tumor?
Yeah, as we discussed, I think this is a challenging area. I do think probably the most easy way to operationalize that idea is if a patient has a change in their biology after getting YL1 treatment, or specifically if they're on targeted therapies that we know cause resistance mutations. Biopsying the progressing lesion to see what is the molecular profile at that time, I think is worthwhile. Just examples of that. For example, currently FGFR inhibitors are approved for FGFR fusion cholangiocarcinoma. We now know that FGFR mutations occur that confer resistance to FGFR inhibitors. There may be other mechanisms that we're looking into as well. A repeat biopsy or a liquid biopsy can give insight into that. There are clinical trials looking at, for example, PARP inhibitors in patients with BRCA mutations. These are still investigational in this setting. We know that BRCA mutations that confer resistance to PARP inhibitors occur. So in the context of that investigational therapy, repeat biopsy is worthwhile. HER2 inhibitors are being explored now for HER2-positive cholangiocarcinoma. HER2 upon treatment with HER2-targeted therapy can be lost. So with a repeat biopsy, I think is worthwhile to see if the tumor is still HER2-positive before transitioning to another HER2-targeted therapy. At least worth considering. So those are, I think, examples, and I think it's hard to do multiple biopsies in multiple areas. So we want to do what's most effective and when is it going to be most informative. But definitely important to look at these questions.
Thank you, Funda. And I think you're still on. This question from Andrea from the audience, is the liquid biopsy science far enough along that we should be advocating for changes to be made in the clinic so that every patient who is on a target therapy have repeated liquid biopsies done at regular intervals through their treatment?
Thank you for that interesting question. So for liquid biopsies, there are several areas of opportunity. I think one is when after a tumor has progressed to look for acquired resistance mutations. I do think that's an area where there's enough data that it's worth thinking about a repeat liquid biopsy. The area that we don't have as much data for is looking at liquid biopsies throughout treatment in a patient who doesn't have any evidence of progression. Now, there's two scenarios where there may be a role for that, but it's not proven. So not, I think currently in only research-only setting. One is getting early liquid biopsy assessment. And there's now many studies in many diseases showing that the amount of circulating DNA goes down if you're responding. Even the early 1 month, 2 week, 1 month assessment can tell if the tumor is responding. That being said, knowing that information has not been shown to change outcomes yet. And without that data, I don't think that's standard of care. Thus, I think we're still trying to prove that is worthwhile doing. Second scenario is looking at the tumor liquid biopsies serially while a patient's on treatment. Again, that's a scenario where we may start seeing acquired resistance mutations, and one thinks that maybe adding an extra drug or changing therapy early may have value, but we don't have any proof that doing that before clinical progression improves survival. Again, we would need clinical trial data to prove that that is worth doing before doing that as a standard clinical practice. Those are two areas that are actively being pursued and, very worthwhile research questions. So stay tuned for that. But I do think, if you've gotten a targeted therapy and you progress after initially benefiting, that's a scenario where there's enough data to order a clinical liquid biopsy in a CLIA platform.
Thank you, Funda. One more question for you. From James, is there a rationale to retreat a patient whose disease has progressed on a HER2 targeted therapy with another HER2 targeting agent?
Excellent question. The answer is yes. HER2 is a very compelling target. I think in the cholangiocarcinoma. Think about breast cancer. In breast cancer, there are 5 different HER2 targeted therapies approved now. And we now know in breast cancer, if patients progress on pertuzumab, trastuzumab, they would potentially still benefit from T-DM1. If they benefit from T-DM1, if they progress on that, they would still benefit from tucatinib combination or would still benefit from, trastuzumab deruxtecan. So similarly, I think eventually we'll get there in cholangiocarcinoma where we'll be able to show that HER2 is relevant as a target for multiple different drugs, and we'll need to identify the best sequence given the safety of each drug, tolerability for each patient, etc. So I do think even if the HER2 targeted therapy, one, didn't work an alternate mechanistically different drug may still work. For example, if you have a signaling inhibitor that patient progresses, I think there's still quite high likelihood that an antibody drug conjugate will work.
Thank you. Thank you. So we'll take this last question because I think we are already up with the time. This is for Patricia. Are you listening, Patricia?
Yes, I'm here. Yes.
So, any idea on the level of heterogeneity in metabolic reprogramming within a tumor? Is it clonal, and how dynamic is the change on metabolic program?
Well, this is a really difficult question to answer because to measure metabolic reprogramming is really difficult. And so I don't think that this has been done. It's a great idea and for sure something really interesting, but nope. I think that, well, in my opinion, the metabolic reprogramming, the change in metabolic rewiring will depend on the patient, will depend on several factors. And now what we are trying to do is just to know how it changes the metabolic rewiring in extra and intrahepatic cholangiocarcinoma, because here the environment is totally different. So first we want to know in cells coming from those tumors, if within the cell there's already differences in metabolic reprogramming. But to know exactly if this is clonal or we don't know it. We don't know it, and it's something really interesting.
Thank you so much. So thank you so much to the audience for a very interesting question, and thank you, the speaker, for great discussion. Keep enjoying the meeting.
Thank you.
Thank you. Bye.
Thank you. Thank you to all of you. Thank you, Sergio. Bye.
Bye.